So I'm going to switch from research to clinical over here and provide a brief overview of the cytogenetic and FISH abnormalities that are seen in hematological malignancies as well as solid tumors. So cytogenetics, as you all know, plays an integral role in the diagnosis and subclassification of leukemias and solid tumors. It helps guide therapeutic decisions. It helps determine response to therapy. It helps in the assessment of progression of the disease and the determination of residual disease. It is an independent prognostic indicator. And at the basic level, it helps us to understand the neoplastic process. It also helps provide genetic counseling for cancers with a genetic basis. Molecular cytogenetics is the result of integration of cytogenetic and molecular techniques. It's mostly complementary to cytogenetics, but it can also be a standalone test. Like cytogenetics, it's important for the diagnostic, prognostic, and management of malignancies. Its advantages are that it's highly sensitive and specific, it's rapid and reliable, and can detect submicroscopic changes. Analysis is possible on interphase nuclei, and this is very critical when we do not have metaphases. As I mentioned, it can be used by itself also to monitor residual disease and sex mismatched transplant recipients to monitor the transplant status. So the resolution of the karyotype is the largest. It provides a genome-wide picture of large structural rearrangements as well as numerical changes. FISH, however, can increase the resolution to 50 to 100 kb. And in addition to microdeletions, it also is able to identify the genome-wide abnormalities, except that it's very specific, so you have to know what you're looking for. Molecular techniques can, of course, detect base pair changes as well. So as of August 2015, a total number of close to 66,000 cases have been reported, with about 10,000 gene fusions identified. However, as you can see, it's very disproportionate. The number of hematological malignancies analyzed is close to 74%, whereas solid tumors is only 26%. And this is very disproportionate to the incidence. The reason being hematological malignancies, there's relative ease of specimen collection. There's no dissociation of cells needed. The culture duration is short. And the chromosomal abnormalities are not nearly as complex. Solid tumors have lagged behind because of mainly technical difficulties and great heterogeneity within the cancer. Given that, mesenchymal tumors have characteristic specific translocations akin to the hematological malignancies. Epithelial tumors are defined predominantly by deletions. Neurogenic tumors also contain mainly numerical changes or amplifications. And germ cell tumors have the ISO12P. FISH analysis is carried out in the clinical lab using basically 3 types of probes. You have the repetitive sequence probe. Not sure why this is not working. The repetitive sequence probe, which are localized to the centromeres and the telomeres. You have the unique sequence probes, which could be dual color, dual fusion, in that the 2 genes are labeled with different colors, and when there is a translocation, you see a fusion signal. You have the break apart probe, in which the 3' and 5' regions of the gene of interest are labeled in different colors. So in a normal situation, you would have the 2 fusion. Pointer pointer's not working. So you have the two fusion signals, and then when there is a rearrangement, you have separation of the signals. In addition, there are other techniques that are fish-based, such as whole chromosome painting, spectral karyotyping or metaphase fish, comparative genomic hybridization, and of course, microarrays. This is the classical Philadelphia translocation between chromosomes 9 and 22. The gene on chromosome 9 is the ABL, and on 22, the BCR. So in a normal situation, you would see, we also include the ASS gene here because deletion at 9q34 confers a poor prognosis in CML. So basically, in a normal situation, you would see the 2 green, 2 red, and 2 aqua signal pattern. When there is a translocation, you have the normal 9 and the normal 22, and the 2 fusions on the derivative chromosomes. That is an example of 3 fusion where you can see an extra copy of the Philadelphia chromosome. And that happens when progression or blast crisis occurs. In myelodysplastic syndromes, you have all of these these are the common abnormalities that you see, and their frequencies. Deletion of 5q when it is present as the sole abnormality has a good prognosis. So does deletion 20q. Monosomy of chromosome 7 or deletion of 7q has a poor prognosis. And trisomy 8 has a good prognosis. This is an example of deletion of 5q and the fish signal pattern. The EGR1 gene on 5q is in red, and you can see that we have only 1 red, but 2 green, which is the control signal on 5p. Similarly, you see 3 copies of the red of chromosome 8, so you have the 3 signals there. And deletion 20q, you have the 1 red because there's a deletion. In AML, the cytogenetic abnormalities are very aligned to the morphology. But the most recent WHO classification of hematological malignancies is based on the genetics of the disease. Translocations that confer a favorable prognosis include the 821 translocation, as shown over there, the 1517 translocation, and the inversion 16. And the fish patterns are also shown there. We already spoke about the deletion. This is the fusion. And this is the example of the break apart where you can see in a normal 16, you have 1 fusion. But because of the inversion and the separation, you see the separation of the signals. Poor prognosis in AML is associated with all of these cytogenetic abnormalities, specifically trisomy 11, deletion, monosomy 5q, deletion, monosomy 7q, DER17, translocation 922 in AML confers a poor prognosis, whereas in CML, it has a good prognosis. In ALL, again, these are some of the specific recurrent translocations and cytogenetic abnormalities that you find. Those that confer favorable prognosis include hyperdiploidy, trisomies of chromosomes 4, 10, and 17, translocations that are associated with the Burkitt, lymphoma, leukemia, 2, 8, 814, and 822, the translocation 1221 and 912. Intermediate prognosis you get with the 119 translocation, the 15q rearrangement, and if you have more than 50 chromosomes. Hyperdiploidy with more than 50 chromosomes. Adverse prognosis, when you have near haploidy, most of the translocations, such as 411, 922, 611, any of the translocations involving the MLL gene, in fact. In CLL, the common chromosomal abnormalities are deletion of 13q, deletion of the ATM gene, and deletion of the TP53 gene, as well as trisomy 12. Deletions of 13q confer a good prognosis. Trisomy 12 is intermediate prognosis, and deletion of ATM and P53 are poor prognostic indicators. In mature B-cell neoplasms as well, you have multiple translocations as well as deletions that are characteristic of these tumors. And most of the abnormalities, other than the 11, 14, confer intermediate to poor prognosis. In B-cell neoplasms, almost 50 to 60% of the translocations involve the IgA gene. Again, these are some of the examples. That's the 814 translocation in Burkitt lymphoma or leukemia. And here you can see it follows the same dual fusion, dual color translocation pattern. So in a normal cell by FISH, you would see 2 green and 2 red. And if there was a translocation, you would see 2 fusion, 1 green and 1 red. In follicular lymphomas, you have the 14-18 translocation. And in mantle cell lymphomas, you have the 11-14 translocation. I just put this slide to show you what the cytogenetic manifestations of amplification are. This is called a homogeneously staining region. And there's another double minutes are another manifestation of this. So you can see we use the MLL probe, and you can see that the amplification is localized to the homogeneously staining region. The IgA gene, which is at the end of 14q32, is retained. So there was no translocation, just amplification of MLL. FISH is also used to monitor bone marrow transplant in sex mismatched cases. We use the X and Y probes, and it can tell us what percentage of the bone marrow cell population is host or donor. In the clinical lab, we use panel FISH. It's a multi-targeted approach in which we use multiple DNA probes. And they are tailored to include probes for the most common abnormalities associated with that disorder. It provides a rapid result because you're looking at multiple probes in the same hybridization. It provides valuable information that helps determine prognosis and diagnosis in patients because especially in the lymphoid malignancies where you don't get that many good metaphases. The percentage of abnormalities detected by panel FISH when cytogenetics fails or is normal sometimes is about 44%, so it's significant. So this is just an example of the panel probe we use for MDS 5, 7, 8, and 20. And these are examples showing the different abnormalities, monosomy or deletion of 5q, monosomy or deletion of 7q, trisomy 8, and deletion 20q. Again, this is just an example of the CLL panel FISH, where we look at ATM, P53, 13, and 12. Coming to the solid tumors, the mesenchymal tumors are a heterogeneous group. And what is really interesting about this group of tumors is that they have very specific chromosomal abnormalities, very specific translocations that are akin to the leukemias and lymphomas. These are etiologically significant, and almost all different histologic types have been shown to have some kind of specific abnormality. So these are some of the common excuse me, translocations, and most of them you can see in close to 90 to 100% of the tumors. Specifically, the Ewing sarcoma with the EWS1 gene that is involved in multiple histologies. Myxoid sarcoma with translocation 1217. Extraskeletal myxoid chondrosarcoma with the translocation 922, and so on. We can detect these translocations in tissue sections as well, formalin-fixed archived tissues. And this is very helpful because sometimes there may not be enough tissue available for cytogenetics or for FISH analysis. So 4 micrometer sections are cut from tissue blocks and FISH analysis is done. This is just an example for the synovial sarcoma with the X18 translocation. The SST gene, you can see it follows the break apart probe logic. So you have the fusion signal, and then when there's a rearrangement, there's separation of the signals. And the same thing for the Ewing sarcoma with the translocation 1122. The epithelial tumors are different in that they are polyclonal with multiple and complex abnormalities. Again, there are very specific abnormalities, and only the kidney tumor has a translocation. We've already heard about HER2 amplification in breast cancers. In bladder cancer, there is a test, the Eurovision FISH, where multiple copies of 3, 7, and 17 in greater than 4 cells, or deletion of 9p21 is considered significant. Neurogenic tumors show amplification as in oligodendrogliomas and neoblastoma with the NMYC. Germ cell tumors, that's an example of the ISO12P. These are some of the familial cancer syndromes and the genes associated with them. To conclude, when a specimen comes in, the question is whether do we do cytogenetics or FISH or both tests. So all newly diagnosed cases should have a cytogenetics analysis done. If specific abnormalities are known, you can perform FISH for that, or if it is a rush case. And then even for triaging between cytogenetics and FISH, all diagnostic cases, or residual or complete remission follow-up samples, and then where it's not indicated are when they're reactive or benign, or if there's no involvement of bone marrow or blood in NHL, and if the diagnostic sample was normal. I'm going to stop there because I think my voice is giving out too. Thank you.